Hello students, myself Ajay Kumar Shivastav. I am senior faculty in botany at Sri Chaitanya. Remember, in the previous chapter we talked about the process of transcription where on a DNA template an RNA is synthesized, a complementary RNA is synthesized, and we saw the difference between prokaryotes and eukaryotes. In prokaryotes or bacteria, directly mRNA is synthesized that can go for translation. But in eukaryotes, initially hnRNA was synthesized and then it underwent capping, tailing and splicing, complete processing. Then it was converted into mRNA. All this was done in the nucleus. Then this mRNA came into the cytoplasm for translation. Now, for translation to occur, the coded information of the DNA has been transferred on the mRNA. That is why it is called as messenger RNA, because it is containing the message of DNA that which protein is to be synthesized. So, this relationship is called genetic code. See, so our next topic is, or next subtopic is the genetic code. So, let us see a specific sequence of nucleotides in DNA or mRNA, because the sequence of nucleotides in mRNA is ultimately determined by the sequence of nucleotides in DNA. So, the specific sequence of nucleotides in DNA or mRNA enables a cell to synthesize a specific protein, means a protein with a specific sequence of amino acids. This interrelationship is called genetic code, that the sequence of nucleotides in DNA or mRNA, they determine what will be the sequence of amino acids in the protein that will be synthesized. Okay. Now, children, see, up till now we studied DNA replication and transcription. It was comparatively easy for the scientists to understand these two phenomena, because both these phenomena are governed by the rule of complementarity. Because you see here DNA replication, if the DNA template has this sequence 5' end ATG, TTT, GAG, 3' end, then the next new strand that will be synthesized, you know that the two strands of DNA are antiparallel and complementary. So, very easy, you can tell of case that other strand will be in 3' end to 5' end orientation, and the sequence of nucleotides will be TAC, AAA, CTC, complementary. So, comparatively easy to understand the phenomena of replication. Likewise, it was easy to understand the phenomena of transcription, because although on a DNA template RNA is synthesized, but this also follows the rule of complementarity. Similarly, if you see the phenomena of transcription, the template DNA is 3' end to 5' end TAC, AAA, CTC. So, the mRNA that will be synthesized, because polymerization always takes place in 5 to 3. So, the template is always 3 to 5. So, mRNA will be, the only difference is that in the RNA, in place of thymine it will be uracil. Okay. So, you see it will be AUG, UUU and GAG, complementary. So, this also is governed, transcription is also governed by complementary. So, this was also easy to understand. But children, the problem was understanding the process of translation, because in translation the template is polynucleotide and the product is protein, means polypeptide, and there is no complementary relationship between the nucleotide and the amino acid, or you can say between the polynucleotide in the polypeptide. So, this was very difficult to understand that how this relationship is governed, but it was confirmed that there is a relationship, because any change in the nucleotide of the DNA or the corresponding mRNA resulted in change in the amino acid in the protein. It means there was a relationship, but how this relationship is established, because there is no complementarity, this was a daunting task for scientists, and this relationship study is genetic code. Now, this became possible due to another RNA called as tRNA. We have studied the structure of tRNA in detail in the previous classes. So, How it became possible? Because you remember tRNA at one end, acceptor end, it has a specific amino acid. So every tRNA carries only a specific amino acid, and at the other end it has a triplet of nucleotides which is called as anticodon. So remember, at one end it has a triplet of anticodon, at another end it has a specific amino acid. So there is a specific relationship between the type of tRNA and the amino acid it carries. Okay. Now this anticodon, it reads the nucleotide sequence on the mRNA. So anticodon of tRNA reads the nucleotide sequence on mRNA. So 3 consecutive nucleotides on mRNA, you can see they code for one amino acid, because a specific tRNA contains a specific amino acid. So we can say that if AUG is the sequence in mRNA, that will be read by only that tRNA that has complementary sequence in 3' end to 5' end, and this tRNA will always carry methionine. So we can say that AUG, these 3 consecutive nucleotides, they code in mRNA, they code for the amino acid F-methionine. So this way a specific relationship between the sequence of nucleotides on mRNA and the sequence of amino acids in the protein was established. Okay. So this is how the sequence of nucleotides in mRNA, this determines the sequence of amino acids in protein, because on the ribosome, when these two will come together, a peptide bond between these 2 amino acids will be formed with the help of peptidyl transferase. Okay. Similarly, a peptide bond between these 2 form. So this way a polypeptide chain will be formed, and the sequence of nucleotides on mRNA will determine the sequence of amino acids in this polypeptide chain, and this is done with the help of tRNA. So 2 points we have to see here. It means we can say that 3 consecutive nucleotides on mRNA code for one amino acid. This is called as a codon. So that is why we say that genetic code has a triplet codon, because 3 consecutive nucleotides are coding for one amino acid. And second thing, you see, there was no direct complementary relationship between the mRNA and the amino acid. Still we were able to synthesize, or the cell is able to synthesize this sequence of amino acids. This is done with the help of the tRNA. It means tRNA, it serves as a bridge and or conduit between the sequence of the nucleotides and the sequence of the amino acid. That is why tRNA is called as an adapter molecule. It works as an adapter between the polynucleotide of the mRNA and the polypeptide of the protein, and this was discovered by Francis Crick. Now, deciphering or decoding the genetic code, this was a very challenging task, and this became possible due to contribution of scientists from many fields. When I say many fields, I mean in physics, organic chemistry, biochemistry, genetics, all they gave their inputs. Okay. Now let us see some properties of the genetic code. First, genetic code is a triplet codon, means 3 consecutive nucleotides in the mRNA code for one amino acid. Okay. Now this is a triplet codon. This was interestingly proposed by a physicist, George Gamow, and for this he used a very simple mathematical proposition. He said that 20 amino acids participate in protein synthesis. These 20 amino acids in different combinations are responsible for production of millions of different types of proteins. Now, he said, but the nucleotides that are coding for these amino acids, they are only 4 types of nucleotides. Okay. In the mRNA, there are only 4 types of nucleotides. Now, if one nucleotide codes for one amino acid, then without any ambiguity, means overlap, without any, only 4 could be coded. Only 4 amino acid could be coded, but the number of amino acid is 20. Now, if combination of 2 nucleotides code for 1 amino acid, then only 16 such combinations are possible, while the amino acids to be coded are 20. It means the minimum should be that a combination of 3 nucleotides code for 1 amino acid. This way you can see 4 to the power 3, 64, means more than 20. So, on the basis of this mathematical proposition, he said that the genetic code should be a triplet codon. Okay. And later on, by the work of Marshall Nirenberg, it was scientifically proved that genetic code is a triplet codon. Now, next property is stop or initiator codon. You see, we just said there are 64 codons. In this, a codon which is AUG, this codon, always from this codon the translation process starts. That is why this codon is called initiator codon. And this codon codes for the amino acid methionine. So, the codon AUG has dual function. First, it acts as an initiator codon, that from here the translation will start, and second, it codes for the amino acid methionine. Okay. So, this is the first start or initiator codon. Similarly, to stop or terminate the translation process also there are certain codons. These codons are called stop codons or terminator codons or nonsense codons. These codons are UAA, UAG and UGA. Now, as I said, there are 64 total codons. This one is the initiator codon AUG, and these 3 are the stop codons. Remember, for these stop codons there are no tRNA, because these codons do not code for any amino acid. They provide the signal for the termination of the translation process. So, remember, for stop codons there are no tRNA. Okay, it means there are only 61 sense codons that code for the different amino acids. Okay. Next property is, children, fourth, comma-less or contiguous, means as you can see, one codon is immediately followed by the other codon. There are no commas, no punctuations in between two codons. This is called as comma-less or contiguous, means one codon is immediately followed by the another codon. Now, next property of genetic code. Genetic code is unambiguous and specific. What does this mean? It says that one codon codes for only one amino acid. Okay, one codon codes only for one amino acid, means there is no ambiguity. So you see what is the advantage? So, if the sequence of nucleotides in the mRNA is known, then the sequence of amino acids coded by this mRNA can be predicted, as you can see here. AUG codes for methionine, we have already discussed. UUU always codes for phenylalanine. UUC also codes for phenylalanine. UAC codes for tyrosine, it will always code for tyrosine, and UCU always codes for serine. So, if we know the sequence of the nucleotides in mRNA, we can predict the sequence of amino acids in the protein that will be produced by the translation of this mRNA. This is called unambiguous. Next, but the reverse is not true. See, genetic code is degenerate. Now, what is the meaning of this? You see, we said that out of the 64 codons, 61 codons code for different amino acids, that is, they are sense codons. But the amino acid types to be coded are only 20. It means for one amino acid, there are more than one codons in many cases. So, some amino acids are coded by more than one codon, and this property of genetic code is called as degenerate. Genetic code is degenerate. Okay. But there are two amino acids that are exception, methionine and tryptophan. These two amino acids have only one codon each. All other amino acids have more than one codon. And maximum degeneracy, if you see the genetic code dictionary, you can see maximum degeneracy is shown by 3 amino acids: leucine, arginine and serine. All these three have 6 codons each. Now students, see, the reverse cannot be done. Here we said if we know the nucleotides on mRNA, we can predict the amino acids in the protein because genetic code is non-ambiguous. But the reverse we cannot do. If we know the if we know the amino acid sequence in the protein, we cannot predict the sequence of nucleotide in mRNA that was translated to synthesize this protein, because you see phenylalanine, it has more than 1 codons. Tyrosine has more than 1 codon. So you cannot say that which codon was present when this tyrosine was incorporated. Serine, there are more than, just now we said that leucine, arginine and serine have 6 codons each. So out of those 6, which was present during the translation, you cannot predict. So if we know the sequence of amino acids in the protein, we cannot predict the sequence of nucleotides in the mRNA that was translated to synthesize this protein. Next property is universal codon. The code is nearly universal. Now what does this mean? This means that from bacteria to human being, in all organisms, the same codon codes for the same amino acid. That from bacteria to human, same codon codes for same amino acid. For example, you see U, it codes for phenylalanine. So in bacteria also it will code for phenylalanine, in human it will also, in all organisms. Now children, the advantage of this is that in biotechnology, in recombinant DNA technology, we transfer the gene of one organism into the other organism. That organism is called as the host. So in the host organism, that gene synthesizes the same protein. Why? It is just because the genetic code is universal, and this question was asked in previous year. While discussing the question, we will discuss this question. Okay, but you know biology is a science of exceptions. So there are some exceptions. In some cases this is not followed. Some exceptions to this rule have been found in the mitochondrial codon, especially you can see yeast and human, and in some protozoans. So one or two such examples I am showing you. See, in universal system, UAA, UAG and UGA are stop codons. We have already discussed. But you see, in human and yeast mitochondria, this UGA, it codes for tryptophan. So this is an exception. Similarly, in universal system, AGA and AGG, these code for arginine, as you can see. But human and yeast mitochondria, these are stop codons, or what you call as the terminator codons. So these are some exceptions. Now we are coming to the last property. Genetic code is non-overlapping. Non-overlapping means that one codon does not overlap the next codon, or you can say that one nucleotide is part of only one codon. You see, this G is part of this codon, the next codon will be here, means this does not overlap. Now how can it be overlap? Suppose if it is like this, the next codon is like this, then G will be overlapping between first and second codon. This is not the case. So one nucleotide is part of only one codon. This is called as genetic code is non-overlapping. So students, with this we have completed all the important properties of the genetic code. Now students, the next important task was to decipher or decode the genetic code, to know that which codon codes for which amino acid. For example, today we know that UUU codes for phenylalanine or AUG codes for methionine. But how we came to this conclusion? See, there was a scientist called Servo Ochoa. He discovered an enzyme called polynucleotide phosphorylase, and now it is called as Ochoa's enzyme. Now what is the advantage of this enzyme? That this enzyme does not require a template. It does not require a template. So it means by using this RNA polymerase, we can polymerize an RNA of defined nucleotide sequence, what we like, desired nucleotide sequence. So by using this enzyme, RNA with defined nucleotide sequence was synthesized in a template independent manner. So we can synthesize an mRNA of our choice, our liking. So this was very instrumental. Why? Because you see, Hargobind Khurana, a scientist of Indian origin, he was a American citizen but Indian origin. Hargobind Khurana, he developed a chemical method by which he was able to synthesize RNA molecules with defined nucleotide sequence. Okay, so this enzyme was very useful in doing this. Now, these were of two types. First, homopolymer. Homopolymer means an RNA which is polymer of only one type of ribonucleotide. So, he synthesized an mRNA which had only one type of nucleotide. So, it had only one codon, one type of codon, UUU. And when this mRNA was used for translation, the protein that was synthesized had only one type of amino acid, that is phenylalanine. So, this mRNA has only one type of codon, UUU, and the protein synthesized by this mRNA had only one type of amino acid, phenylalanine. So, this proves that the codon UUU codes for phenylalanine. And students, this was the first codon to be cracked. Next one is copolymers. Here you can see a pair of nucleotides are repeated in a definite sequence. Okay, so UG, it is repeated. As a result, we are getting two types of codon, UGU and GUG, UGU, GUG, this sequence. Now, when this mRNA was subjected to cell-free protein synthesis, the protein that was synthesized, it had two amino acids, cysteine and valine, alternating each other, cysteine, valine. So, this proved that UGU codes for cysteine and GUG codes for valine. Okay. So, this was instrumental in deciphering the genetic code. Now, all these cell-free system work was done by Marshall Nirenberg. Okay. So, he was very instrumental in deciphering the genetic code. His cell-free system of protein synthesis finally helped all the codons to be deciphered. So, this is all regarding deciphering of the genetic code. Next thing is mutations and genetic code. You see, mutations are very useful in understanding the relation between DNA and gene. Okay. DNA functions as gene because any change in the DNA, mutation in the DNA, it causes change in the protein, as a result change in the phenotype. So, this relationship can be easily understood. Now, point mutation. Change in single base pair in DNA is called as point mutation. It is a type of gene mutation. Now, gene mutations are of two types. First is substitution. There is no increase or decrease in the number of nucleotides in the DNA, but one nucleotide pair or base pair is replaced by another one. This is called as substitution. And a very good example of substitution due to which a genetic disorder arises is sickle-cell anemia. In hemoglobin, there are two chains, alpha chains, two types of chains rather, alpha chain and beta chain. In the beta chain, in 6th position, normally the amino acid is glutamic acid. Why? Because in DNA, the codon is GAG-CTC. Okay. This undergoes transcription to produce the mRNA codon GAG, and GAG codes for glutamic acid. When there is mutation in the DNA, see mutant, then you see it becomes CAC, means the CTC of DNA due to mutation changes into CAC. So, after transcription, the codon in the mRNA changes to GUG, from GAG it changes to GUG, and GUG codes for valine. So, in the beta chain of hemoglobin, at the 6th position, glutamic acid is replaced by valine, and this valine causes sickling of the RBC and many other problems that you have already studied. Okay. So, this is a good example of substitution mutation. Let us see another type of gene mutations called as frameshift mutation. These arise when there is either insertion or deletion of one or two nucleotides. Remember, one or two nucleotides. Okay, and this can be very well understood by this sentence. See, this sentence, each word represents a codon, because you know a codon consists of 3 nucleotides. Suppose these are the 4 codons and this sentence is Ram has red cap. Now if we insert B between has and red, you see Ram has, up till here it is correct, but after that the entire reading frame is changing, BRE, DCAP. So after this all the codons change, that is why it is called as Frameshift mutation. Now let us see insertion of 2 nucleotides, insertion of BI. Ram has, up till here no problem, but after that you see BIR, EDC, AP, all are changing. So insertion or even deletion of 1 or 2 nucleotides causes change in the entire reading frame. That is why it is called as Frameshift mutation. But you see, if we insert a complete codon, a triplet or multiples of a triplet, then you see Ram has big red cap. So there is insertion of one word, but the entire reading frame is not changing. So remember, insertion or deletion of a triplet or multiple of a triplet does not cause Frameshift mutation. It causes either addition of one or few amino acids in the protein or deletion of one or few amino acids in the protein. So students, with this we have completed the portion of genetic code, and this portion is very important for the NEET examination point of view, because just last year 2019 2 questions were there in the NEET paper related to genetic code part. Let us see these 2 questions. So NEET 2019, under which of the following conditions will there be no change in the reading frame of following mRNA? Remember, we said Frameshift mutation means change in the reading frame takes place when there is insertion or deletion of 1 or 2 nucleotides. Insertion or deletion of a triplet will not change the entire reading frame. If there is insertion of a triplet, means 3 nucleotides, then 1 codon will increase or 1 amino acid will increase, but rest of them will remain as such. Similarly, if we delete a triplet, means 3 nucleotides, then there will be loss of 1 codon, as a result loss of 1 amino acid, but all other codons will remain as such. So let us see the options. This is the mRNA given 5' end to 3' end, AAC, AGC, GGU, GCU and AUU. Okay. Insertion of G at the 5th position. This is 1, 2, 3, 4, 5. If you insert G, then all the codons after this will change, means this is not our answer. Second, deletion of G from 5th. Similarly, 1, 2, 3, 4, 5. If you delete this G, all the codons from here will change. So this is not our answer. Third, insertion of A and G at 4th and 5th position. Same, if you 1, 2, 3, 4. If you remove these 2, then also all the subsequent codons will change. So this is also not our answer. Fourth, deletion of GGU from 7th, 8th and 9th position. Okay, let us see. 1, 2, 3, 4, 5, 6, 7. If you remove this GGU, then you see AAC, AGC, GCU, AUU. All other codons have not changed. Only 1 codon has decreased. It means this removal of GGU or a complete triplet will not cause any change in the reading frame. So our answer is option number 4. Clear? Let us see the next question. Which of the following features of genetic code does allow bacteria to produce human insulin by recombinant DNA technology? Okay, why a particular protein is synthesized by a particular gene in whichever organism we insert? it is due to the universal nature of the genetic code, that is from bacteria to human, same codon codes for same amino acid. So, you see our answer will be genetic code is nearly universal. Redundant means useless. They mean to say that the genetic code is useless. No, genetic code is very useful. So, children, with that we have seen these two questions also that were asked in Neet. With this we come to the end of this lecture. I hope you enjoyed the lecture. Please be very safe in your homes.